Hello, everyone, and welcome to our presentation on applications of nitrogen vacancy centers in diamonds. My name is Amanda Jenkins. I will be discussing an overview of nitrogen vacancy centers, as well as quantum sensing and computing. A nitrogen vacancy center is just a nitrogen atom that has an adjacent vacant site within a diamond. These are very common within diamonds, and scientists are able to observe that electron spin echo with great accuracy. It's more isolated. It has weak coupling with its surrounding atoms within this lattice, creating a more isolated spin. And scientists are still able to influence that in order to get different readings based on what type of quantum mechanics they're studying. An important note is they're also solid state at room temperature, which is very important when studying quantum mechanics and applies to a wide variety of different uses because of this. Um, they're very stable and can be replicated easily. Scientists have had trouble in the past creating that kind of isolated environment that they're able to sort of replicate across different applications. Some of the topics we'll be covering today are quantum technology, biofluorescent labelings, temperature sensing, magnetic field measurements, and things related to the biomedical field. The first topic we'll be discussing is quantum sensing. This more so helps in all of these kind of applications. It's kind of the fundamentals. It's them sensing the actual magnetic fields that is created by those electron spins. They can, as you can see, they can detect at really low levels, like almost the individual atom within ourselves, which really helps with those biological processes that we'll see later. This process involves recording the individual spin of each nitrogen vacant center, and then using a mathematical process called covariance to observe the correlation between these magnetic fields and how they're interacting with each other. So quantum sensing, a fundamental application, can be used across multiple industries, um, which leads us, this quantum sensing also leads us to quantum computing. Quantum computing is a relatively new thing. It involves, it's built upon the classic computer. It has the ability to solve problems faster and with more complexity than regular computers. This is due to a thing called qubits. A regular computer is using bits in order to process information, but these qubits use quantum mechanics in order and quantum sensing in order, and they're able to exist in multiple states that are able to process information much faster than bits can. And they're really good at calculating those properties and physicalities of quantum mechanics in nature. So they'll have applications from pharmaceutical research to finance to climate change solution strategies, really important with things like machine learning and auto industries, things computers can relatively do already, but with more complexity and faster with this ever evolving technological world that we're living in, this is a very important application.
My name is Kayana Morris, and another one of our topics is biofluorescent labeling. Biofluorescent labeling uses fluorescent tags to observe and trace a biological molecule or structure. The purpose is to enhance the study of cellular and molecular processes and is often combined with advanced imaging techniques. The applications, like how we use this in everyday life, is cellular imaging, molecular sensing, and quantum biosensing. In cellular imaging, NV centers can be used to label specific proteins to allow for precise imaging. In molecular sensing, NV centers detect nanoscale magnetic fields, allowing for tracking of biochemical changes. And in quantum biosensing, it's used to detect single molecules and cellular interactions. As you can see in the picture up here with the human, you can see how the fluorescent drugs were put into there so that they could detect and get a picture of the actual the actual molecules that they were looking for. Um, in the rat down here, you can see how they put a bacteria in the rat and they were able to trace the changes that it done into his body while it was inside. The advantages and disadvantages are they're non-invasive, so it's safe for live cell imaging and it's least invasive to biological samples. It's durable, so NV centers can maintain fluorescence under many conditions. And it's multifunctional, so it works in all kinds of different settings, like quantum sensing, high resolution imaging, and tracking molecular dynamics. The disadvantages are it's really hard to to target specific biomolecules because the NV centers are a little bit bigger than what they would use in a different kind of test. The fluorescent stability in biological environments. NV centers are stable under standard conditions, but the fluorescence can still be impacted by pH, ionic strength, and temperature changes. And one of the most disadvantage is the cost because it requires diamond synthesis, which is much more expensive than like their standard dyes or standard tests that they would use.
Temperature sensing uses the defect in nitrogen vacant diamonds to measure temperature by nanoscale sensing. Now, what does this mean? This means that the defect in nitrogen vacant diamonds holds a specific type of power that allows for diamonds to be used as a very impressive conductor. Diamonds naturally are already great conductors due to their ability to conduct heat properly, but not conduct electricity, which makes it perfect for temperature sensing as it does not attract and get disrupted by the magnetic fields around it, but it is able to concentrate on conducting the heat and being able to withstand high temperatures all the way up to 600 degrees Kelvin and to 0 degrees Kelvin. The accuracy of the temperature sensing is so precise that it is regarded on the nanoscale, which means you can get a very accurate depiction of heating and melting points. The accuracy for these diamonds is around 98%. Temperature sensing can be applied for different vectors of life, such as physics, biology, and chemistry. Normally, these properties are useful for when it comes to nanotechnology and biological processing. As with anything, there will be pros and there will be cons of temperature sensing. However, in my opinion, I believe that the pros outweigh the cons when it comes down to the accuracy and how it is able to be applied to life. The pros would include stability, which is powered by diamond's ability to have very strong covalent bonds, and it is a very stable molecule. Along with this, it has long spin coherence, which means that it is less likely to make errors and is less likely to pick up on any type of complications. Next would be the optical properties. This is because diamonds themselves have a very high refractive index. However, there are negative consequences, and that is because the diamonds themselves for temperature sensing have to be very particular. So it has to have the nitrogen vacancy inside the diamond, as well as it has to be a particularly small diamond, because the larger the diamond is, the more sensitive it will be to the magnetic fields around it, and the sensitivity to the magnetic fields can obscure the results. Along with this, the diamond has to be of higher quality because that, of course, can also obscure the results, which can make it very difficult to obtain for smaller facilities and for different businesses that are trying to look into temperature sensing.
As NV centers are highly sensitive to magnetic fields, they can be utilized as imaging tools in medicine. MRIs use magnetic fields to create an image of the body, but NV centers could bring out a higher resolution than MRI scans. Due to the high spatial resolution of NV centers, they can push past the microscale most MRIs allow, giving us an even more detailed image of the human body and its cells. As most cells and tissue have their own magnetic field, NV centers can react to them, notifying us of abnormal cell growth. One method involves nanodiamonds containing NV centers being injected into the cells of interest. These magnetically quote-unquote labeled cells can now be differentiated from healthy cells. Magnetic concentration of the cell's proteins can allow rapid testing and early detection. At a nanoscale, researchers would be able to pinpoint mutating cells and cancer biomarkers. As discussed, NV centers have been suggested in helping researchers target abnormal cell growth and immune responses. A relevant example is the use of NV centers in quantum diamond microscopes to measure the concentration levels of interleukin-6. This protein has been tied to the body's immune responses, such as helping B lymphocytes produce antibodies, causing fever, and maintaining metabolism. Yet elevated levels of interleukin-6 can be due to inflammation, autoimmune disorders, cancer, and COVID-19. An immunoassay called LumenAssay can help scientists tag interleukin-6 proteins by using beads that are coated in a specific antibody that will stick to a cell of interest. This method has been used to identify multiple diseases, but its turnaround time is long and only manageable in large batches. As a solution, it was suggested that quantum diamond microscopes that use a synthetic diamond chip doped with NV centers be used to measure the IL-6 concentration levels. This was tested in Boston with patients in hospitals for COVID. 35 patients with COVID were included in the study. Magnetically labeled plasma samples were placed in the microscope and their magnetic fields were measured using the NV centers. When hit with the green fluorescent light, NV centers emit a red fluorescence that can be used to make an image of the concentration of IL-6. It was shown that higher levels of IL-6 correlated with more severe cases of COVID. It was found that this method is just as accurate as the average LumenAssay, but also faster and can be utilized in smaller sample sizes. This more rapid turnover time can aid in making quick clinical decisions and can help providers manage patients better. Its cost is also lower than LumenAssay, which can benefit the economic strain hospital labs can have in times such as the pandemic. This makes it more available to patients who are admitted to emergency departments and can shorten hospital stay time and hospital cost.